Hey guys, welcome back to my channel. In this video, we are going to continue our topic that is transport across cell membrane. I have been making a video from long back, so if you want to check those video regarding the transport, so you can better understand this topic, the link is in the description. You can go check it out. In the last video, I have been telling that transport across cell membrane is divided into two: passive transport, where no energy is required, and the second is active transport, which is an energy required process. In passive transport, we talked about its two subtypes: diffusion and osmosis. Later onwards, we divide diffusion in simple diffusion, facilitated diffusion, and non-ionic diffusion. Now, coming to our today's topic, that is active transport, which is an energy based process that is divided into two types: primary and secondary. I will talk about the same in a while. So, first, a brief introduction on active transport. Active transport is a movement of molecules against the chemical, electrical, or electrochemical gradient. So, you can say it is uphill transport. Just assume this like swimming against the water tide in a river. You are going against the flow. So, obviously, if you are going against the flow, you will require energy to conquer that flow. So, it is an energy based process, and we get this energy by breakdown of high energy compound, that is adenosine triphosphate, or simply ATP. Active transport exhibits saturational kinetics, meaning active transport reaches a saturation point due to the limited availability of carrier and supply of energy. As you know, active transport along with the facilitated diffusion are carrier mediated transport, meaning they require carrier protein for transport of molecules and substance across the cell membrane. So, two types of active transport are primary active transport and secondary active transport. First, primary active transport. In primary active transport, the energy is directly derived from the breakdown of ATP or some high energy phosphate compounds. The mechanism operates for ion pump. Ion pump hydrolyzes the ATP to ADP and uses this energy for the transport process of the molecules. The important primary active transport processes are sodium potassium pump, potassium hydrogen pump, and calcium pump. So first, sodium potassium pump, or you can say sodium potassium ATPase. It's very important, and we are going to learn this in detail compared to other pumps. So sodium potassium are transported across the cell membrane using a carrier protein called sodium potassium pump or sodium potassium ATPase. This pump transport sodium from inside of the cell to the outside of the cell, meaning from ICF to ECF, and potassium from the outside of the cell to the interior of the cell. The point of discussion for this pump are structure of the pump, mechanism of its action, function, regulation, and the clinical part, that is applied physiology. So discussing the first one, that is the structure of pump. As you know, sodium potassium pump is a carrier protein, and it is made up of two globular protein subunit, namely alpha subunit and the beta subunit. The alpha subunit is concerned with the ion transport, meaning the transport of sodium and potassium, while the beta subunit's functions are not clear, but you can say it stabilizes the alpha subunit. Alpha subunit has 5 different binding sites, 3 on its intracellular site and 2 on its extracellular site. From the 3 intracellular binding site, one is for the 3 sodium ions, one is for the ATP, and the one site is reserved for the phosphorylation. From the 2 extracellular site, one is for the potassium ion and the other is for the albumin, that is a steroid hormone that inhibits the sodium potassium pump. On the next slide, you can see the structure of the pump. The pink colored diagram shows the alpha and the beta subunit, alpha on the right and the beta on the left. But you can also see that the inner portion or the intracellular part near the sodium binding site has ATPase activity on both alpha and beta subunits. Now let's learn the mechanism of action of sodium potassium pump. The step first is binding of 3 sodium ions and 1 ATP on its respective binding side will activate Atpase enzyme that hydrolyze ATP to ADP, releasing high energy phosphate bond. This change will bring a conformational change due to the phosphorylation and will eject 3 sodium ion extracellularly, meaning out of the cell. Subsequently, 2 potassium ion binds on the outer side that will lead to dephosphorylation, meaning the phosphate group will remove, and this in turn cause the protein to return to its normal or original conformation, and the potassium ion are ejected inside the cell. So you can conclude during one cycle of phosphorylation and dephosphorylation, 3 sodium ions are pumped out and 2 potassium ions are pumped in, and only 1 ATP is hydrolyzed. So now what is the exact function of sodium potassium pump? So first you can say it causes electronegativity inside the cell, meaning 3 sodium ions are put out and 2 potassium ions are moved in. So clearly loss of one positive charge ion is there. That will increase negativity inside the cell, which is very important for the cell signaling. The second, it prevents the accumulation of sodium ion inside. An exit of potassium from the cell along their concentration gradient will maintain an ionic equilibrium. Third, it prevents accumulation of water inside the cell by maintaining the ionic concentration on both the sides of cell membrane. And if it fails to do so, it will lead to swelling of cell followed by its rupture. So the next is regulation of pump. In the regulation, we will talk about how the activities of sodium potassium pump is increased by some of the hormones or how it is decreased by the inhibitor. So activities of sodium potassium pump is increased by cyclic AMP, or you can say CAM, in the renal cortical collecting tubules of mammals. Second, by diacylglycerol, thyroid hormone, aldosterone and insulin increases the activities of sodium potassium pump, while the activities of sodium potassium pump is decreased by or at low temperature, lack of oxygen, dopamine and orbin, which is an inhibitor. Talking about the last part for the sodium potassium pump is the abnormalities or applied physiology, you can say. So the first is reduction in either its number or concentration of sodium potassium pump in the myocardium is associated with cardiac failure. Second, reabsorption of sodium in the renal tubule is associated with hypertension. So the second pump in the primary active transport is potassium hydrogen pump or potassium hydrogen ATPase. So it is present in the parietal cell of gastric gland and the renal tubules. It is a P-type ATPase similar to sodium potassium pump. It is also a P-type of ATPase. If you want to know about type of ATPase, comment down below. Surely I will make a video on type of ATPase also. It has two subunits. The sodium potassium pump will have two subunits, alpha subunits and the beta subunits. The alpha subunit will contain catalytic sites of enzyme and forms a pore through the cell membrane that allows the transport of ion, while the beta subunit will stabilize the alpha subunits. So the potassium hydrogen ATPase transport one hydrogen ion in exchange of one potassium ion. So as the ion pump transport ion against the concentration gradient using energy derived from the hydrolysis of ATP. This phosphate transfers power a conformational change in the enzyme that helps derive ion transport. The hydrogen potassium pump present in the parietal cell transport hydrogen out of the cell into the lumen of gastric gland, and potassium ion from the lumen are transported inside the cell. And the potassium hydrogen pump present in the cell of distal tubule of nephron secretes large amount of hydrogen ion into the tubules to eliminate them from the body. Now, what are the function of potassium hydrogen pump? First, acidification of stomach content. Second, it activates digestive enzyme, that is pepsin. And third, it maintains acid base balance. The next part of discussion here is inhibition and some clinical points. Inhibiting gastric gland secretion by blocking the ATPase activity is very common clinical intervention used in diseases including dyspepsia, peptic ulcer diseases, and gastroesophageal reflux diseases. Two categories of drugs are used to inhibit the hydrogen potassium ATPase activity. The first is a hydrogen receptor antagonist like cimetidine, common name Tagamet, inhibits the signaling pathway that leads to the activation of ATPase. Cimetidine blocks the histamine from signaling the H2 receptor located on the parietal cells of gastric gland. As you know, these cells are responsible for hydrochloric acid secretion and secretion of intrinsic factor. So blocking this ATPase activity will help in the reduction of HCl secretion. As you know, in gastroesophageal reflux disease, the HCl reaches up to the esophageal level, causing burns and harm to the esophageal part. So it will help in reducing those effects. The second category is proton pump inhibitors, PPIs, like omeprazole or Prilosec, the common name, that directly binds to the hydrogen potassium ATPase and inactivates the pump. Moving on towards our last pump for the primary active transport, that is calcium pump. Like the sodium potassium pump, it also operates with help of carrier protein which has ATPase activity. They are responsible for active transport of calcium outside the cell for maintenance of extremely low concentration of calcium in the ECF. The calcium pump plays a crucial role in proper cell signaling by keeping the intracellular calcium concentration roughly 10,000 times lower than the ECF concentration. And failure to do so is the one of the major cause for muscle cramps. The calcium pump is not only present on the cell membrane, which exclude the calcium out of the cell, but it is also present in some cell organelles such as sarcoplasmic reticulum in the muscles and the mitochondria of all the cells. This calcium pumps move calcium inside the cell organelles. Function of calcium pump. Calmodulin is a calcium dependent protein which require calcium for its activation. Calmodulin plays an important role in smooth muscle contraction. Second, calcium is important for the cell signaling. Third, it acts as an intracellular messenger. So for this video, it's all about the primary active transport. In the next video, we'll talk about secondary active transport and its type, co-transport and counter-transport. Thank you for watching. Follow me on Instagram where I post questions regarding the video content. See you in the next video.